Hey, Anatomy 1 students, what's going on? We are on our second video about the introduction to the nervous system. Last time we covered the structure of a neuron, like this guy right here and this guy right here. And what I want you to check out is that these two neurons are meeting right here. So I've got a neuron, here's its cell body, here's its axon. The axon comes out and ends in its axon terminals. And where the axon terminals are meeting the cell body of this neuron or the dendrite of this neuron, this is called a synapse. Like this junction here is called a synapse. It's the way neurons connect. It's the point at which neurons meet and communicate. It could also be the junction between a neuron and an effector cell, like a muscle cell or a gland cell. And by the way, notice that the flow of information is going to be in this direction. There's an error already, but I want to add to it. The flow of information is going to be in this direction, okay? In this direction. That's the direction of the flow of information. Because of that, this neuron over here is said to be the presynaptic one, and this guy is said to be the postsynaptic one, with information, communication traveling from pre to post. All right, let's zoom in on a synapse. We've done that here. So here is my presynaptic neuron. This is its axon up here. This is its axon right here. Probably the telodendrion of an axon to be exact. Now down here is the axon terminal. And the axon terminal is coming right up against the cell body. So here's the soma, the cell body of another neuron. Now this guy is my postsynaptic neuron. Notice there is a gap, a space between the two neurons. That space is called the synaptic cleft. Space is called the synaptic cleft. Now I also want to point out that the axon terminal is filled with little membrane bags. And those little membrane bags are called synaptic vesicles. They're filled with chemical signals that are called neurotransmitters. So what's going to happen is when the electrical signal comes down the presynaptic axon, it's going to cause these synaptic vesicles to release their contents. They're going to do some exocytosis that releases the neurotransmitters into the synaptic cleft. And the neurotransmitters in the synaptic cleft are going to diffuse and bind to these receptors here, which then allow the postsynaptic neuron to do something. So electrical signal called an action potential comes down the axon, prompts the release of neurotransmitters from our axon terminal of our presynaptic neuron. The neurotransmitters then bind to receptors on the postsynaptic neuron and cause a response in it. Okay, so here we see just another example of a synapse. This is my presynaptic neuron. This is my postsynaptic neuron. Their meeting point here is a synapse. Here is the cleft between them. When the nerve impulse, the electrical signal or action potential gets down here, the synaptic vesicles undergo exocytosis, releasing neurotransmitters into the cleft. The neurotransmitters then bind to these receptors, causing and creating a response in our postsynaptic neuron. Now, as we said earlier, synapses are not just between two neurons. Right here we see an axon of a neuron coming down. Here's a telodendrion, some more telodendria coming out. Each telodendron is ending in an axon terminal, and the axon terminal is coming right up next to our muscle cell here. And this junction between the axon terminal and the muscle cell is another example of a synapse. Now, in particular, this kind of synapse is known as a neuromuscular junction because it's a synapse, a meeting, a joining between a neuron and a muscle cell. Okay, good. Let's now that we've got the basic anatomy of a neuron in terms of its soma, its dendrites, its axons, and its synapses, let's look at the 3 different kinds of neurons. There are 3 different structural classifications of neurons. There are multipolar neurons. This is a multipolar neuron right here. Notice his cell body has multiple processes. This is a bipolar neuron right here. Notice his cell body, his soma, just has 2 processes. And my final dude over here is a unipolar neuron, which is a single short process that splits rather quickly, coming off of his soma. Let's talk about all 3 of these. Multipolar neurons are going to have 1 axon and multiple dendrites. We've been looking at multipolar neurons so far. This is a multipolar neuron over here. Most neurons in my body are multipolar neurons, including motor neurons, which are the ones that are going to send commands to effectors, as well as interneurons, which are kind of like neurons that talk just to other neurons. More on these guys later on. Estimates vary. Some people say, 90%, 95% of your body's neurons are multipolar. But it's the majority, and it's a significant majority. Now, this is a bipolar neuron. It's got a soma here, a cell body. It's got that 1 axon, of course, and a 1 dendrite that comes out. Then that dendrite branches into many branches at the end. So 1 dendrite that gives off a bunch of branches, and then 1 axon. This is going to be a bipolar neuron. They're only found in 2 places. They're found in my olfactory epithelium. So they're very much involved in smelling, and the olfactory epithelium is in the superior nasal cavity. These guys are also involved in the sense of vision. They're found in the light-sensitive organ of your eye, which is the retina. Here we have a nasal cavity, all sorts of good bones that we know about in here. And at the top up here, this is where I'm going to find some bipolar neurons. So in this superior nasal cavity, so you guys know what bone this is, by the way? We're zooming in on it right here. It's got a bunch of holes in it. That bone is the ethmoid bone. One of my all-time favorites, definitely. It's got those olfactory foramina in it. And these yellow guys here are olfactory neurons. They are in this superior olfactory epithelium of the nasal cavity. There's the cell body of it right there. And we got the dendrite right there with many branches. And then on the other side of the cell body, we got the axon, which is then going up through the olfactory foramina to talk to some relay neurons up here. So these are bipolar neurons involved in your sense of smell in your nasal cavity. Now, as I said a moment ago, we also have bipolar neurons in our retina. This is the eyeball here. Anterior is on this side, posterior is on this side. In the back of the eye, there's this light-sensitive organ called the retina. And we're zooming in on the retina here. So these are all the different cell layers of the retina, including the photoreceptor cells that respond to light. And then adjacent to them, these reddish guys here, these are bipolar neurons. These are bipolar neurons involved in the signaling going from your eye to your brain, letting your brain know about the light that has struck these photoreceptors. Okay, unipolar neurons. The cell body, the soma, has a single process coming out of it, and that single process connects to the axon. Well, this is weird. This is really weird. Check this out. Here are the dendrites over here. The dendrites fuse together to make the axon. Then the axon stretches from one edge to the other. These would be the telodendria over here and the axon terminals. And then along the way, the cell body is just kind of like a little side thing on the axon. Now, the axon is divided up into two parts. The part between the dendrites and the cell body is called the peripheral process. The part between the cell body and the telodendria and the terminals is called the central process. So these guys are sensory neurons. They're going to be sensory neurons dealing with touch, temperature, pain, and proprioception. Remember, that was our awareness of like joint position, limb position. And we'll take a look at some more of these guys in a second. Obviously, the central process is the one that is going towards the brain or spinal cord, going towards the central nervous system. The peripheral process is the one coming from the periphery. All right, here is a spinal cord right here. Here's a multipolar motor neuron with his cell body, his dendrites, his axon. Here's a multipolar interneuron, a neuron between two neurons here. And the blue guy is a sensory neuron. Here are his dendrites fusing together to make his peripheral process of his axon, which goes past the cell body here. Then it becomes the central process, which is then going into the spinal cord, branching and ending in its axon terminals, which are synapsing with this interneuron. So sensory neurons like this are unipolar neurons. By the way, notice there's a cell body right here. Of course, there would be many cell bodies clustered together here, not just one. And this spot where there are a bunch of cell bodies together, remember, that is called a ganglion. So a little review right there. Okay, those were my structural types of neurons. I've also got three functional types of neurons. We've kind of seen all three already. I've got sensory neurons, interneurons, which are also called association neurons, and I've also got motor neurons too. Sensory neurons are mostly unipolar. Some are bipolar, like the ones in the retina and in the superior olfactory epithelium of the nose. Sensory neurons are afferent, meaning they're taking information from the body to the CNS, to the brain and the spinal cord. This information can be about so many different things: odor, sound, light, taste, touch, pressure, stretch, pain, temperature, levels of ion, levels of gases, and even more. Here is a sensory neuron right here, this blue guy. Sensory neuron taking information from the periphery in towards the spinal cord, towards the CNS. Now, motor neurons are multipolar neurons that are either going to directly or indirectly control effector cells. And remember, effector cells are generally muscle cells or gland cells. There are actually two kinds of basic motor neurons that are known as the lower and the upper motor neurons. Lower motor neurons are found in the brain and the spinal cord, and they directly synapse with skeletal muscle cells. Upper motor neurons are found in the brain, and they're going to signal the lower motor neurons in the spinal cord. So two different types here. Let's see if we have a picture here. Here I have a motor neuron in my spinal cord going directly to an effector, to a muscle or a gland. Now in this picture, I've got some upper motor neurons, a pair of them represented by these black lines here. And these upper motor neurons are coming out of the brain down into the spinal cord, where they're then synapsing with these red guys. And these red guys are lower motor neurons, and they are going to skeletal muscles. All right, so those are our two types of motor neurons that we've got. And finally, we've got the interneurons. These are typically multipolar neurons, and they're found entirely within the CNS. As their name implies, interneurons, they're going to communicate between sensory and motor neurons. They're also called association neurons, and that has to do with their ability to process and store information. So in this picture right here, we have a sensory neuron, the red guy, stimulated by the heat of the candle there. The sensory neuron takes information in. Notice it's a unipolar neuron. Sensory neuron then synapses with the interneuron. It is processing that information. The interneuron is then telling the motor neuron to tell the muscle to contract so we can withdraw from that painful stimulus. So those are my three functional types of neurons. What I want to talk about now is how neurons communicate. So we know that communication between neurons happens at a synapse, where the presynaptic neuron releases chemical signals called neurotransmitters, and those chemical signals bind to the postsynaptic neuron. But what I want to talk about now is how does information travel from one end of the neuron, like from the dendrites, all the way to the axon terminals? And the answer is it's electrical signals that do that. Electrical signals are going to take information from the dendrites to the soma, from the soma to the axon hillock, and from the axon hillock to the axon terminals. Now in my neuron, the electrical signals are these waves of change. A wave of change means something changes in one spot, which causes it to change in the next, and then in the next, and then the next, and next. Like when you when you knock over a row of dominoes, the first domino knocks over the second, which knocks over the third. The dominoes themselves aren't moving very much, but there is a wave of movement along the line of dominoes. We'll talk more about this, and we'll explain what we mean by membrane potential. Now, the electrical signals that happen in dendrites and in the soma are called graded potentials, whereas the electrical signals happening in the axon are known as action potentials. Okay, let's establish what I mean when I say membrane potential. The membrane potential is this state in our neuron where there's an electrical difference between the inside of the cell and the outside of the cell. So in this example here, we are actually measuring the electricity inside and outside of the axon here. We're using a device called a voltmeter, and we can see the inside of the cell is more negative than the outside. So about a difference of 70 millivolts. The inside is at negative 70 millivolts compared to the outside. And this electrical difference is known as a membrane potential. Now, why do we have a membrane potential? We have a membrane potential because of three membrane proteins. One of them we've met already, the sodium-potassium pump. This is a sodium-potassium pump right here. Sodium-potassium pump is in the membrane of our neurons. So throughout our neurons, dendrite, soma, axon, there are lots of sodium-potassium pumps. Here's the phospholipid bilayer of the membrane. Here is the protein, the sodium-potassium pump. It's constantly breaking down ATP to move 3 sodiums out and 2 potassiums in. Notice that in doing so, I'm moving more more positive charges out than I'm moving positive charges in. So I got 3 pluses going out, but only 2 pluses going in. That's going to make the interior more negative, which is what we saw in our voltmeter here. Okay, so the sodium-potassium pump helps make the interior more negative. Also, throughout my neuron, in my dendrites, in my axon, in my soma, in the phospholipid bilayer, there are these proteins called potassium leakage channels. Potassium is always leaking out. As potassium leaks out, that is going to make the interior of the cell more negative. And again, notice that the interior of the cell is negative compared to the exterior. Finally, we've got some sodium channels in the membrane as well, allowing some sodium to leak in. If there were no sodium channels in the membrane, then the membrane potential would be something like negative 90 millivolts. But because some sodium leaks in, the membrane potential, the electrical difference, is such that the cell interior is at negative 70 millivolts. Okay, so that's what we mean when we say the cell has a membrane potential. Now, oftentimes we'll refer to this membrane potential as the resting membrane potential. So that negative 70 is the resting membrane potential. And that's the membrane potential when the neuron is not sending any electrical signals. Another way to describe our neuron, because it has a membrane potential, we say it is a polarized cell. The word polarized means there's a difference between the interior and the exterior, and that is a difference in charge, with the inside being slightly negative compared to the outside, and that slight negativity has a value of negative 70 millivolts. So our neuron is said to be polarized. This is going to matter in a moment, as we shall see. Okay. Whoops, I should probably get rid of that. There are 2 fundamental ways the resting membrane potential can change. It can change one way, which is called depolarization, and then it can change another way, which is called hyperpolarization. So it can depolarize, it can also hyperpolarize. Depolarization is an increase in the membrane potential. So notice that the membrane potential is at negative 70. That's our resting value. Nothing's going on. But if some sodium ions rush in to the neuron, and this is the point we're measuring here. So this spot right here is negative 70 at rest. Then if at that spot some sodium ions go into the neuron, that'll make the interior more positive, and our membrane potential is increasing, meaning it's going up here from negative 70 to negative 60 to negative 50. That's an increase. And that's called a depolarization. Notice that if we're depolarizing, we're making the inside more positive. If we make the inside more positive, that means there's less of a difference between the inside and the outside. Another way of saying that we're lessening a difference is we're depolarizing. So D like meaning removing, polarization meaning the difference. Okay. The other thing we got to notice is if we depolarize, well, we got to get back to that normal resting value. And that return to the resting value is called a repolarization. So in this case, some sodium ions entered our neuron right here, causing this particular spot to depolarize, meaning its membrane potential got more positive. But we're going to have to bring that back to normal. And that return to normal is called repolarization. Okay. The other thing we could do is we could hyperpolarize. So again, at this spot in the neuron, we're at rest, we're at negative 70 millivolts. And then either some potassium ions leave or some chloride ions go in. Notice chloride is negatively charged. In both of these situations, either pluses exiting or negatives entering. That's going to make the inside of the cell more negative. So negative 70 becomes negative 80, negative 90, negative 100. And if the inside is even more negative than normal, it'll be even more different than the outside. So a greater hyper difference polarization. So those are the basic fundamental ways that I can adjust my membrane potential. I can either depolarize it or I can hyperpolarize it. And we talked about it changing because ions were either going in or out of the cell. Ions like sodium, potassium, and chloride. Now we got to figure out how do we get ions to go in and out, and how do we regulate that? And we do it with two varieties of ion channels. There are ion channels called ligand-gated ion channels and voltage-gated ion channels. This guy here, this channel protein in our phospholipid bilayer, is a ligand-gated channel. When a neurotransmitter, the red guy here, binds to him, he opens up, allowing ions to pass through, in this case, to enter the neuron. And when ions are going into the neuron, or if they were exiting, that would impact the membrane potential. Because we either got positive charges going in or out, or negative charges going in or out, and that's going to change the electrical difference. So that's a ligand-gated channel. The other type of channel is a voltage-gated channel. It doesn't open in response to binding to a neurotransmitter. Instead, it opens by changing shape when the membrane potential reaches a special value called the threshold potential. And that special value is typically negative 55 millivolts. Now, a really important fact we got to be aware of is that voltage-gated channels are only found on axons. Voltage-gated channels are not found on neurons. They're not found on dendrites of neurons, not found on the soma of neurons either. Voltage-gated channels are only found in axons, and this will become important later on when we are dealing with the signals that are traveling in a neuron. All right, we're about half an hour in, and I am going to call it quits on Nervous System Intro lecture number 2. So we will end here. We'll pick this up next time. We'll finish the nervous system intro, knock it out in the next video. All right, see you later. Bye.